Hey everyone, my name is Kashish Kundu and I will present my topic that is in-silico analysis of transcriptomic responses to UV and ionization radiations in human cells. So, to understand the human response to DNA damage, we use the microarray data from a dataset that is GSA in 1977, and we use microarray to measure the transcriptional responses of the genes to ionization radiations IR, which is represented in red in my samples, and uh, UV, that is ultraviolet radiation, is represented as blue in my samples. Along with one more sample is there, that is the mock, which I represented with green. So there are many genes that are involved in our known DNA damage response pathways for cell signaling, proliferation, and many other metabolic pathways in our body. However, the majority of the genes are involved in unexpected pathways with functions in signal transductions, the RNA binding and editing, and synthesis or degradation of the protein. So I won't go into the methods of how this data was actually extracted, but I'll just tell the brief about it that about 10,000 genes were analyzed in this sample, which were collected from total of 15 healthy individuals without any history in cancer and were immortalized with Epstein-Barr virus. So, to obtain a genome-wide portrait of the transcriptional response to DNA damage in human cells, oligonucleotides, microarray was used to measure the responses of the genes in cell lines for these different individuals, and the cell lines served as replicates and allowed us to identify responses that were independent of other variations among the different individuals. Now, the pipeline that I followed here, I identified gene identification. I selected a data set. So for selection of data set, how I selected it is I was interested in knowing how cancer we get skin cancer by UV radiations, basically sun rays. So how we humans are getting infected. The what is the effect of sun rays on our skin? So this was the particular paper which was quite intriguing. So I downloaded the series matrix file and I removed the unwanted information and selected the gene expression table. After grouping the data into mock, IR and UV, I mentioned how I colored code them. This was the coding: mock green, red IR, and blue UV. Then I went for PC analysis of the data, and I was experimenting with my PCA analysis, so I went for three different types of PCA by comparing the mock with IR and UV separately and all three together. Then I went for statistical analysis and calculated the p-value, q-value, that is p-adjusted value, and log FC value or the fold change value. For all this procedure, I use the T-Bioinfo platform, and this platform is provided by Pine Biotech. And for running these pipelines, I use this platform, and you can see there are so many pipelines that are here, and it gives us a wide area of analysis here. For PCA, I use unsupervised analysis and the utility box also has PCA. There are some different parameters which you can follow here. You can always go to the T-Bio server and look into it. Then this is the data set. This is the dataset, and you can see the dataset here. This is the series matrix table. I removed this one, and id ref I changed it to id. I made some relevant changes into it and removed the pre-documentation information that was given. Then I went for grouping the data. I grouped them as IR, mock, and UV. Later on, I went to PC analysis. This is the T-Bioinfo platform. Here you can see the result output that I could get. I have the text file for the PCA. I have a zip folder which has all these files and obviously the PDF plot. So you can see here my PCA at first was not that good because, oh sorry, because there were many the samples were forming a carpet, so there was not a good distinct grouping was not there for you for all the three samples or the individual samples. So I gave it a thought about it, and I thought that after sorting this data and filtering the data, maybe I could come back and again do the PC analysis and see if I get some relevant information. For p-value formula, I used equals to t dot test followed by the array 1, that is the mock sample, array 2, and the parameters. Array 2 is the IR samples that I have. For p-adjusted formula, I use the formula as is equal to, and then the AF144 is the p-value. When selecting the p-value, later I gave the max of the counts divided by a individual p-value, individual count of that p-value, respective p-values. Then for fold change or log FC formula, I use the log of average of the mock samples divided by the average of the IR samples. Now in the same way I did it for UV samples. Once this was done, now I was left with how now, as you all know, for microarray data we have the probe IDs, we don't have the gene names and the gene IDs. So my task was to get the gene names and the gene IDs. So I've used David here. David is the database for annotation, visualization, and integrated discovery. These tools are powered by the comprehensive David knowledge base, and that is built upon the on David gene concept, which pulls together all the multiple sources of functional annotations. Once these sources are collected together, they have various different tools you can use. And once these one of the tool is gene name batch view. I use that for changing the probe IDs into relevant gene IDs, not actually changing, for merging them. So I selected the sorted data. Now for sorting the data, how I sorted, I selected the Q value as less than 0.05, and the log FC value I choose it between 1 and 1 or -1. So then I selected the data, a whole file, and I went to the upload section and put I just copy and pasted the file here and selected the some parameters changes here and hit run. After running, I got the David IDs, which are actually the probe IDs with the gene name and the gene IDs. You can see the gene IDs are written in the bracket. So now the task that is left is merging the data using R. So after merging the data, now after merging the data, I'll go for pathway analysis using links tool. Now links is also it also has a wide range of tools for performing pathway analysis and statistical analysis and whatnot. It's a huge server. So in future prospect, after pathway analysis, I'll go for the result and interpretation of the data. um, so these are the two references that I have used till now. This is the paper from where this data was taken, and all the research work that they have, how they extracted the gene that is also given here, and how they process the microarray data, that is also given here. This is the data, and this paper has how it's about what you all need to know about DNA damage and repair regarding microarray data. Thank you from my side. This is the work that I have done, and probably I'll go for pathway analysis in the future and definitely share the result with you guys again. Thank you.